Okay, so welcome to this next video on the Vogelstein model of colorectal carcinoma. So we have so far got to the point where we have this intermediate adenoma, which is made up of these early adenoma cells, which just have two mutations in their APC genes, which are both loss of function, and also these pink cells, which have both the two loss of function mutations in APC genes and also the gain of function mutation in the KRAS protein. What's going to now happen is one of these pink cells, which has these 3 mutations already, is now going to suffer 2 more mutations, 2 loss of function mutations in the SMAD4 genes. Okay, so let's discuss this then. Right, we need, in order to understand this process, we need to understand how the transforming growth factor beta pathway works. So, transforming growth factor beta is a growth inhibiting molecule. So let's say this represents transforming growth factor beta. Transforming growth factor beta, which is often usually denoted TGF beta. So this is transforming growth factor beta, okay, and TGF beta for short. Okay, right, so transforming growth factor beta binds to the transforming growth factor beta receptor, which is a dimer of 2 receptors. So let's denote that here. Okay, so it's made up of a type 1 transforming growth factor beta receptor and a type 2 transforming growth factor beta receptor. So let's say here on the left is our type 2 transforming growth factor beta receptor, and on the right is our type 1 transforming growth factor beta receptor. So let me colour this in. So we'll have transforming growth factor beta in this pink colour here, okay, and then we'll have the type 2 transforming growth factor beta receptor here in green. Okay, right, and then we'll have the other type 1 transforming growth factor beta receptor in this turquoise colour on the right here. Okay, so let me label everything up. So this is the type 1 TGF beta receptor here on the right. So this is type 1 TGF beta receptor. Okay, and this on the left is the type 2 TGF beta receptor, and they dimerize together to form the active TGF beta receptor. So this is the type 2 TGF beta receptor. Okay, right, so these the domains that I've drawn on the intracellular aspect of these 2 receptors, those are serine-threonine kinase domains. So they add phosphate groups onto serine and threonine residues. So these are serine-threonine kinases. Okay, so let me just remind you of the structure of a serine and a threonine amino acid residue. Okay, so these are serine-threonine kinases. Right, so the structure of a serine or a threonine amino acid then. So, let's begin with the basic amino acid structure. So here's the amino group here. Then the amino group comes off the alpha carbon here, which also has a hydrogen coming off it, and a carboxyl group down here. Okay, so that's the basic backbone of an amino acid. Okay, now also off the alpha carbon you have the R group. Now in the case of serine, the R group is a methylene group, with then a hydroxyl group coming off that methylene group, like so. So this is the amino acid structure of serine. Okay, now let's look at the structure of threonine. So threonine again has the normal backbone of the amino acid structure. So here's the alpha carbon with the amino group coming off it, and the carboxyl group down here. Okay, and then also off this alpha carbon you then have the R group, and in the case of threonine the R group is like so. So you have this carbon and the hydroxyl group again, and then, but also off this carbon you have a methyl group like so. So that's the structure of the threonine amino acid. Right, okay, so the way in which you add phosphate groups onto these serine and threonine amino acids is you add phosphate groups onto the hydroxyl groups here. Excuse me. And basically, if I draw a phosphate group here, so here's a phosphate group, it consists of a phosphorus atom with a double bond to an oxygen there, and then these two hydroxyl groups coming off it, and also a single bond to an oxygen which has a negative charge then, because it's acquired an electron ionically from some other chemical species. And then what happens is that you can link this phosphate group to the hydroxyl group of the R group of either serine or threonine via a condensation reaction. So basically you can take this hydrogen off this hydroxyl group, and you can take the hydroxyl group off the phosphate group, and you can combine those together to make water. That's why it's called a condensation reaction, because it produces water. Then you combine this oxygen to the phosphorus atom there, and that forms the link between the oxygen and the phosphorus atom. Okay, and that's the way that you phosphorylate a serine residue. And the same thing applies for the threonine residue. It's exactly the same, except you've got this methyl group there. Okay, so these serine threonine kinases are going to bind phosphate groups onto the hydroxyl groups of serine and threonine. And generally they don't do it just from inorganic phosphate like I've shown here. Instead, what they will do is they'll use ATP to provide the phosphate group for this phosphorylation process. And in so doing, the ATP will be broken down to ADP, adenosine diphosphate, so it will lose one of its phosphate groups, which will be bound onto the hydroxyl group of either the serine or the threonine residue. Okay, so basically what happens is when transforming growth factor beta, or TGF-beta, binds to the transforming growth factor beta receptor, the serine threonine kinase of the type 2 TGF-beta receptor becomes active. So this here becomes active, and it is now capable of phosphorylating serine and threonine residues on the type 1 TGF-beta receptor. So that's what it's going to do. It's going to phosphorylate serine and threonine residues on this type 1 TGF-beta receptor, specifically on the serine threonine kinase residue of the type 1 TGF-beta receptor. So it's going to stick a phosphate group on here. That's going to cause the activation of this serine threonine kinase of the type 1 receptor. So basically what's going to happen is the TGF binding activates the serine threonine kinase of the type 2 TGF-beta receptor, which then phosphorylates and activates the serine threonine kinase of the type 1 TGF-beta. Now, basically, what happens next is that a SMAD protein comes along, or a SMAD protein comes along. So let's denote this like so. So in comes what's known as an R-SMAD. And basically an R-SMAD, so this is an R-SMAD. R-SMAD can either be SMAD3 or it can be SMAD2. And I don't know why I said SMAD3 before SMAD2. But basically this can either be the protein called SMAD2 or it can be SMAD3. So sometimes instead of writing R-SMAD, I might just write SMAD2 / SMAD3 to denote that it can be either SMAD2 or it can be SMAD3. Right, okay, so this R-SMAD, or this SMAD2 / SMAD3, which I'll denote in this purple colour here, okay, this is going to come over to the type 1 TGF-beta receptor, and the serine threonine kinase of that type 1 TGF-beta receptor, which is now active, is going to phosphorylate the R-SMAD and change its conformation. So let's show the R-SMAD phosphorylated now and conformation changed. Okay, what's going to happen is this R-SMAD, which is either SMAD2 or SMAD3, is then going to form a complex with a protein known as SMAD4. Okay, so let's draw this down here. So, here is our SMAD2 or SMAD3 protein, which has this phosphate group attached to it here, and basically it's going to form a complex with SMAD4, which is initially in this inactive state, but when it binds to the SMAD2 / 3, which is phosphorylated, it's going to again take this sort of active shape here. So let me colour everything in and label everything up. So in purple here, is our SMAD2/3, or our R-SMAD, which is now phosphorylated. Okay, and in orange we'll have, in orange here, this is our SMAD4 protein. Okay, so here's SMAD4, and I think I'll highlight the linker between them, it looks nicer. Okay, and there's SMAD4 here then. This is SMAD4. So what you get basically is this complex between the, oh sorry, not SMAD2, SMAD4. You get this complex between the R-SMAD, which is either SMAD2 or SMAD3, and the SMAD4 protein. Now this complex then goes into the nucleus of the cell. So this is going to go into the nucleus of the cell, and it's going to act as a transcription factor, basically. So here's the nucleus of the cell, and basically it's going to upregulate the expression of certain genes which are involved in stopping the progression of the cell cycle. Okay, so overall, the genes which it's going to upregulate by binding to the promoter regions of, so let me just draw this here. So the R-SMAD SMAD4 complex will bind to the promoter region of certain genes, and basically it will make it more likely that the RNA polymerase enzyme is going to bind to that promoter region, and therefore it's going to increase the transcription of that gene. So it's going to increase the transcription of those target genes, and the specific two target genes that SMAD4 R-SMAD complexes target are P15 and P21. And these proteins both stop the cell cycle. So these stop division. P15 stops the progression from the G1 phase to the S phase. P21 stops progression from the G1 to the S phase. It stops the S phase. It stops the M phase. It stops everything, basically. So these stop the cell cycle. Stop the cell cycle, or arrest the cell cycle is the fancy terminology for it. So they're going to stop the cells dividing. So the transforming growth factor beta pathway is a way of stopping cells dividing. If you spray TGF-beta on cells which are dividing, they should stop dividing, because the P15 and P21 levels in their cytoplasm will go up, and they will halt the cell cycle where it is, basically. Okay, so what would happen if you had a complete loss of function of SMAD4? If both of your SMAD4 genes have loss of function mutations in them. So again, you have two SMAD4 genes, which we'll denote here. So here's one, here's the second SMAD4 gene. What if you got a loss of function mutation in both SMAD4 genes? Then you would have no functional SMAD4. And if you had no functional SMAD4, then you wouldn't be able to form these R-SMAD SMAD4 complexes. And even though the transforming growth factor beta was binding to its receptor and activating these R-SMADs, if you don't have any SMAD4, it can't form this complex, and therefore it cannot activate the transcription of P15 and P21, which are the key proteins which actually arrest the cell cycle and stop cells dividing. Okay, so this pathway would be completely broken if you had loss of function in both of the SMAD4 genes. So SMAD4 would lead to oncogenesis. It would lead to more likely that you develop cancer, because it's if you were to knock both of them out, you'd be more likely to develop cancer, basically, because you're going to get over-division, because it's involved in stopping division. So SMAD4 is another example of a tumor suppressor gene, basically. So one which prevents growth, and therefore if you want to develop cancer, you need to lose function in both of those genes. You need both of them to suffer loss of function mutations. Okay, right, so the next stage then, so let me get another piece of paper and I'll summarize the next stage in our Vogelstein model of colorectal carcinoma. So, if this is the colonic epithelium here, Right. Then we've seen that so far in the Vogelstein model, what has happened is you started off with a single cell here, which was unlucky enough to suffer 2 loss-of-function mutations to its Adenomatous polyposis coli gene. That led to beta-catenin going up in this cell, and it divided too much. So it made a whole mass of cells which were genetically identical to itself, and which all had this double loss-of-function mutation in APC. So you ended up with a whole population of cells which all had this genetic defect. Now, in one of those cells, in that huge, great population, I want to stress, I've drawn, how many cells have I drawn here? 9 cells. You know, you'd have millions, loads of them. This would be a massive, great population of cells. In one of them, one of those cells just happens to be unlucky enough to then get a gain-of-function mutation in KRAS on top of its 2 loss-of-function mutations in APC. So it then starts dividing even more, basically, because it's not only got that loss-of-function mutation in APC, in both APC genes, but also it's got this gain-of-function in KRAS, and that means that it can divide even more than these other cells, or even more than these green cells. So basically it produces a clone of genetically identical cells, it produces a population of genetically identical cells which all have both the 2 loss-of-function mutations in the 2 APC genes, and also this gain-of-function mutation in the KRAS gene. Now, the next stage is that one of these cells in this pink population of cells, which so far has 2 loss-of-function mutations in APC and 1 gain-of-function mutation in KRAS, is unlucky enough to suffer 2 mutations which are loss-of-function mutations in the 2 SMAD4 genes. So it gets 2 loss-of-function mutations in its 2 SMAD4 genes. On top of that gain-of-function mutation in KRAS, so 1 gain-of-function mutation in KRAS, and the 2 loss-of-function mutations in APC, which it already has. So one of these pink cells undergoes this next mutation, and let's draw that now in blue. Now, so it's already got these 2 things which are promoting growth. Now it's got this loss-of-function mutation in SMAD4, which means that the transforming growth factor pathway, which was inhibiting growth of this cell, is now gone, basically. So it can divide even more. So it divides and divides and divides and divides and divides. So it produces daughter cells, which then divide again, and before long you have got an entire population of cells which is genetically identical to this blue cell here, basically. And that, again, it's an even larger population of those than you even had before of the pink and the green ones. So let me colour these in blue. Right, so this stage of the cancer now, well, it's not cancer yet, it's still not cancer, it's a benign tumour still, but this is now known as a late adenoma, basically, when you have these cells which have 2 loss-of-function mutations in APC, a gain-of-function mutation in KRAS, and 2 loss-of-function mutations in SMAD4. So this now is the late adenoma stage. Late adenoma. Okay, right. So again, I want to stress this concept of intra-tumor heterogeneity. If I was to now look at the cells in this late adenoma, you would see 3 different types of cells. In reality, you'd probably see even more, but in my model, you'd see 3 different types of cells. These ones which have all of these 5 mutations, ones which just have 3 of them, and ones which have 2 of them. So you've got different populations of cells which are different levels of the way along to becoming cancer, basically. So this is the concept of intratumor heterogeneity. Okay, right. So we'll call it there for this video. In the next video, we'll see the final mutation that happens in order to turn these blue cells into cancer. So the next mutation is going to again occur now in these blue cells, and that one of those blue cells in that population will then have the final mutation, which is then going to lead it to grow out of control, make a huge number of genetically unstable offspring, which can then become invasive and destructive and metastatic and cause cancer. And that final mutation that's going to occur is in p53.